Hi everyone, this is Miss Segovia and I'm going to walk you through your notes on Punnett squares. So this is just a real basic intro. There's a lot more to this, but we got to start somewhere. So as always, set this up Cornell style. Make sure that you have your extra wide margin going down the left hand side of your paper, and then save a little space at the bottom for your summary. So you can go back later and add that information and have a better understanding. So our question that we're going to address in this one, put this at the top. Do all tall plants have the same genotype? And if you remember from our last lesson, genotype is referring specifically to those alleles. And so if we're looking at tall, we'd probably use the letter T. So does that mean all tall plants are big T? Or could there be something else? So let's start really quickly with the history. Reginald Punnett. If you ever wonder why something has kind of a strange name, like a Punnett Square, it's probably because that was named after someone. So Reginald Punnett came up with Punnett squares. So that's one of our vocab terms, so let's go ahead and underline it. And he did this to predict the proportions of possible genotypes. And offspring. If you remember, Gregor Mendel saw that there were trends and saw that there were numbers involved, and Punnett later on came in and said, hey, here's a method we could use to predict those. And so if we set up a Punnett square, we draw it out. We're just going to do a simple monohybrid cross, which means we're only looking at one trait, and those are 4-square Punnett. When we put the information on there, we put one parent's genotype on the top. Usually it's the father, but it could always be different. So it would be do like a big T and a little t. And so this is usually the male parent. If you put the female up on top, that's okay too. It's like a tradition, that the way I was taught. So male parent genotype. I'm spelling things wrong. G-E-N-O type. So we would put that up there. So if it's a male parent with a female parent, this would actually be the sperm up here. So sometimes we can draw it with a little tail, so remember that's the sperm contributing to the offspring. And then we would do the female genotype down the side, and so those are usually the eggs. So this would be the female parent genotype over here. So male genotype, that's the sperm, if it's a sexually reproducing, and then this would be the eggs down the side, the possibility. And so when we get into meiosis, this will make a little bit more sense. And so again, this is a Punnett square for a single trait. We can also study more traits at a time, doing what's called dihybrid and trihybrid crosses, but those Punnetts get a little bit more complicated, and we'll see that for next time. So let's do a practice Punnett here, doing monohybrid cross with a heterozygous parent. So those are words, I know it sounds like I'm really intelligent and using big words, but monohybrid means we're just looking at one trait here, so monohybrid cross. And they're telling us that the parents are both heterozygous. So that means if we're looking at their genotype, we know that they've got one dominant gene and one recessive gene for their alleles. And so let's do pea color, for example, and the color yellow. So let's say we're taking two parents with peas that are yellow. So each parent, since it's heterozygous, would be big Y, little y, and we would cross that with another parent that's got the big Y, little y. And why am I drawing these circles at the front? Because I want to use some colors. Since we're representing colors, let's do that. I've got my color pencils right here. And so I'm going to color these in as yellow. So if they're heterozygous, in Mendelian complete genetics, the parent would show the dominant trait. And so our dominant trait in our example here today is yellow. And so let's go ahead and draw out our Punnett Square. So we're going to make a 4 square since it is a monohybrid. We're looking at just one trait. And so we would have the male, or one parent, we don't know which one's male or female, but one genotype up here. So it has the big Y, and then the little y goes over the other square, because it has the option of giving its offspring the dominant gene or the recessive gene. The female, or other parent, would go down the side. And since that one has the same genotype, we would do the same thing here. They can give either the dominant gene or the recessive gene to their offspring. And again, in meiosis, we'll see this a little bit more clearly. So when these two cross, we're going to get big Y here. And so what's that offspring going to be? Well, if it's got the dominant trait and the dominant trait is yellow, that offspring will be yellow in color. This one here is going to be our heterozygote offspring. And so since it has the dominant trait, it'll also be yellow. Here is also heterozygous and going to show the dominant trait. So again, yellow. But this one here is inheriting both the recessive genes. And so in my example that we're using here, this is kind of the traditional P cross. If they don't inherit the yellow genes, that means they end up being green. And every problem that you work, they'll usually give you that information in advance as to what the dominant recessive is. So don't worry if you don't know automatically what color it's going to be. This is just my little thing here. So our little narrative, if we are crossing two pea plants that are heterozygous for seed color, which means they are big Y, little y, they will produce offspring that are, all right, now here's where we get the ratios. So let's do our genotype ratios first. So for genotype, we're looking at the big Y's and little y's. We're ignoring the color right now. And so our genotypes, if we look here, we have out of our 4 squares, 1 out of the 4 is going to be homozygous dominant. So I'm going to have 1/4 equals homozygous dominant. That's the big Y, big Y. 2 out of 4, or 1/2, are going to be heterozygous, which is big Y, little y. And then 1 out of the 4 is going to be homozygous recessive, which means that it's going to be the little y genotype. So this traditional ratio, if we ignore the 1/2 here and we just keep it as the force, will be a 1:2:1 ratio of my homozygous dominant to my heterozygous to my homozygous recessive. And that's usually the order we present it in, with the homozygous dominant, heterozygous, and homozygous recessive. If we want to look at the phenotype ratio, this is what we actually see. And so in our phenotype ratio, we have 3/4 are going to be yellow and 1/4 is going to be green. So this is a 3:1 ratio on our phenotype and a 1:2:1 ratio on our genotype. And so you can be questioned on those as to, what's the phenotypic ratio? Phenotypic, let's put that in our questions. What is phenotypic? Phenotypic ratio. Phenotypic is another term that we could use for phenotype. And so they're just asking you what is the phenotypic ratio. In this case, it would be 3:1. So this is our real basic monohybrid cross. So let's go ahead and box this up nice and neat because we're going to do another section here. Let's look at a cross between homozygous plants and what that would look like with our ratio. So let's do a monohybrid cross. Again, looking at just one trait. But with homozygous parents. And so in this one, what we're doing, if we're crossing homozygous, that means both parents are going to have just a single genome. So they're not going to be heterozygous like here where they were big Y, little y. What we're doing is we're crossing big Y, big Y. So if we saw that in the pea color, with a plant that is little y, little y. And so if yellow is our dominant color, that means this parent is yellow and that this parent is green. And so when we do the cross here, we're going to end up with our Punnett Square looking like this. We're going to draw our square out with the 4. And we're going to have big Y, because that's what this parent has right here. And then little y, little y. So this offspring is going to be big Y, little y. Here we'll have big Y, little y, big Y, little y, big Y, little y. Now if Y is the dominant color, that means even though these all had a green parent who gave them the gene for green, because they received the gene for yellow, all the offspring will appear yellow. And so our ratio for the genotype is that we have 4 out of 4 are big Y, little y, which is heterozygous because we have the 2. Our phenotype is also 4 out of 4 for yellow. So we would see 100% of each one. So let's write that out in sentence form. So a cross between a yellow homozygous plant, again that was big Y, with a heterozygous, oh gosh, no, sorry, with a homozygous, I was thinking ahead to what the offspring was, a cross between a yellow homozygous plant with a homozygous recessive green plant, which is little y, only produce heterozygous yellow offspring. So we would have 100% being the heterozygous and yellow. So I apologize for my little mistake there. Hopefully you guys will catch that and make yours look a lot nicer. So our Punnett Square is just, again, real simply, this is a monohybrid cross looking at just one. And so we can also do, and here let's put a little line to separate this out, a dihybrid cross would have a 4 by 4 square Punnett. Sorry, I'm really doing good. Punnett Punnett, because it is analyzing two traits. So if we're doing that kind of cross, that would be like if we're looking at, say, a plant that was tall and yellow, cross with a plant that's, say, short and green. And it's a lot more complicated, and we will take some time on those, but just want to make sure you see the difference between a dihybrid being a lot bigger than our monohybrid. So if we go back up to our leading question, do all tall plants have the same genotype? Well, in our example today, we use the color yellow and green, but if we were to treat it as tall and short, would all tall plants have the same genotype? Not necessarily. They could be heterozygous, big little, or they could be homozygous dominant, big big. And so here we could put our little answer is no. They could be big T, or big T, little t. Just depends upon the situation, and really the best way to tell is to look at their parents. If they had a tall and a short parent, they're most likely heterozygous. If both parents are tall, well, they could be heterozygous or homozygous. We would have to find out what the parents were and look at the grandparents and go back in the line. But we'll do some more of that later in the year. I don't want to get you guys too confused now. For the short of it, that is your notes on Punnett squares. Thanks.